Alright, so obviously as humans we know that we need to obtain nutrients from the food that we eat and oxygen from the air that we breathe, but what is the purpose of that on a cellular level? Well, both of these things are involved in cellular respiration, which is the conversion of chemical energy to usable energy in the cell. And cellular respiration is used in almost every single organism on Earth except for a couple of bacteria. And this even includes autotrophs such as plants, which while they can make their own food, they need a process to be able to break it down and form energy. So, here's the overall reaction. We have glucose plus oxygen leads to carbon dioxide, water, and energy in this usable form called ATP that we'll get into shortly. So let's start with glucose. Glucose is a simple sugar that we can obtain from eating carbohydrates and digesting carbohydrates. So some foods that are very rich in carbohydrates are bread, pasta, and rice. And the reason why runners will actually carbo load right before a race is because these foods are rich in glucose. And as the reaction goes on, you'll see that glucose forms ATP, which is a form of energy, so they won't run out of energy during the race if they carbo load. And then obviously everyone knows that we inhale oxygen. Oxygen is going to be used to drive some electrons and drive the process of making ATP, but we'll get into that. And then it actually has two byproducts that we exhale, which are carbon dioxide and water. Um, so carbon dioxide, we all know that we exhale carbon dioxide, but you might not have known that we are also exhaling water vapor. And this is apparent on a cold day, where when you exhale the water vapor, where while it's warm enough inside of your warm body to remain a gas, as you breathe it out, when it react or well when it gets exposed to the cold air, it's going to condense and form a cloud. And then you have ATP, which is the usable energy in the cell, and this is a basic structure, and we'll talk about that later. So let's see why can glucose be broken down into ATP? Well, it's important to look at its chemical energy. As you can see, glucose has quite a lot of bonds, and chemical energy is energy that's stored in bonds. So glucose, while it has a ton of bonds, the byproducts don't have nearly as many bonds, being the water molecules being very small and the CO2 molecules. And this can be represented by this type of graph. So glucose, it's very big, has a lot of energy, and it's kind of like a block tower where it's unstable with all that energy. And by breaking apart into CO2 and H2O, as you can see, it's at a lower energy level in comparison to this big glucose molecule, just because of their size. But you also might notice that these aren't equivalent. These two energy levels, the CO2 and the H2O, are not equivalent to the glucose molecule. But with the addition of ATP, that makes the product's energy level equal to the energy found in the glucose. So let's talk about how ATP actually works. Um, so ATP, also known as adenosine triphosphate, has three phosphate groups, and as the phosphate group breaks off, it releases free energy. So this once again is chemical energy being converted into actual energy, free energy, heat energy, and this can be used. This can be used for all sorts of cellular processes, whether that be moving your body or importing things into the cell, exporting things into the cell, or out of the cell. Um, and that's all by donating one of their phosphate groups, and then this becomes ADP, di, meaning 2-phosphate. So you might be asking yourself if ATP is just another way of converting chemical energy into usable free energy, then why don't we just break down glucose to power all of our processes? And the answer for that is that ATP doesn't have nearly as much chemical energy as glucose does. By powering your cell's processes directly with glucose, that'd be kind of like powering your car with a nuclear reactor. You'd be getting a lot more energy than you'd need, and it'd be wasteful and inefficient, and it just wouldn't be good. Another important thing to note about this reaction here, the ATP to ADP, is that you can also go the other way around, and you can reattach a phosphate molecule or phosphate group onto this molecule and form ATP again, and that's what happens in cellular respiration. So where does all this take place? Well, in eukaryotic cells, it's going to happen in the mitochondria, and in prokaryotic cells, it's going to happen in their cytoplasm. But for this video, we're going to be focusing on eukaryotic cells for the most part, so let's talk about the structure of the mitochondria. The mitochondria has an outer membrane and an inner membrane, and in between these two, they have the intermembrane space. And then you can see that the inner membrane also has several folds. These are called cristae, and they're going to increase the surface area of the inner membrane. And then on the inside of the inner membrane, we have the mitochondrial matrix, so we'll talk about these later. Uh, Cellular respiration consists of three steps. The first one being glycolysis, which occurs in the cytoplasm. The second being the citric acid cycle, which occurs in the mitochondrial matrix. And the third being the electron transport chain, which happens in the matrix and in the inner membrane. So, the first step is glycolysis. The cell is going to take in a glucose molecule, and then within the cytoplasm, it's going to go through several changes. So glucose, ultimately a 6-carbon molecule, will convert into two 3-carbon molecules of pyruvic acid, or pyruvate. Then, this also generates 2 ATP by combining ADP with phosphates. It's also going to give electrons to NAD+, and this will form NADH. And this molecule has a lot of chemical energy and is somewhat unstable, and we'll harvest this energy later on in the process. It's also going to form 2 protons here. So that diagram didn't show it, but along with all of these other products, this also releases 2 H2O, which we breathe out, like I talked about before. So we've gone from glucose to pyruvate in glycolysis. We've formed NADH and it's carrying electrons, and we've also generated a little bit of ATP. Alright, let's talk a bit more about that pyruvate that we formed in glycolysis. So to enter the citric acid cycle, which is the next step in cellular respiration, pyruvate needs to convert to acetyl-CoA. And so acetyl-CoA has 2 carbons, unlike pyruvate's 3, so it loses a carbon in the form of CO2, and that, once again, is one of our byproducts. And this molecule of pyruvate is going to react or combine with this coenzyme A to form the acetyl-CoA, and in this process, it also donates electrons to NAD+, forming NADH. So now we have the citric acid cycle, and it's also called the Krebs cycle, either or. This is the reaction we just did, pyruvate to acetyl-CoA, and as acetyl-CoA goes through, as you can see, it's going to lose another 2 CO2. And then so for the 1 pyruvate with 1, 2, 3 carbons, it's going to lose 1 carbon here, 1 carbon here, and 1 carbon here, all in the form of CO2. So all of the original carbon from pyruvate is gone. Also, it's going to give electrons to NAD+, forming NADH along the way a few times. It's going to form some ATP as well, and then along with the NADH that is formed all throughout this cycle, it's also going to form FADH2, which is a molecule that's very similar to NADH. It's carrying electrons, and it will be able to drop them off in the electron transport chain. So this is the overall reaction. Oh shoot! This is the overall reaction for the citric acid cycle. And this is for 2 acetyl-CoA. So this is for the 1 glucose goes to 2 pyruvate goes to 2 acetyl-CoA. And the acetyl-CoA is after the pyruvate already formed some NADH. So just keep that in mind. It also forms some CO2. And I'll make things more clear towards the end. But so the 2 acetyl-CoA is going to give off 4 CO2. It's going to create 2 ATP. And it is also going to add electrons to these 2 molecules, forming 6 NADH and 2 FADH2. So, we have gone from pyruvate to acetyl-CoA. Acetyl-CoA goes into the citric acid cycle, generating a little bit of ATP, and then it has also formed NADH and FADH2, and these molecules will go to the electron transport chain to drop off their electrons, along with the NADH that was formed from glycolysis. Okay, so we just did the Krebs cycle in the mitochondrial matrix, and up next is the electron transport chain. And the electron transport chain itself is actually on this inner membrane here, and that's the importance of these cristae, these inner folds. Increasing the surface area allows there to be more electron transport chains, thus making it more efficient. So this is the electron transport chain right here, and it looks pretty intimidating, but I swear it's not that bad. So we just did the Krebs cycle in the matrix. Here's this inner mitochondrial membrane. This is the actual electron transport chain itself. It's a series of proteins, and this is the intermembrane space. So what's going to happen is that electrons from NADH and FADH2 are going to travel down these proteins. And they're drawn down these proteins because they're attracted to this oxygen. This is the oxygen that we breathe in, and it has a very, very high affinity for electrons. And without this oxygen that we breathe in, then this process would stop, and so would the generation of ATP in this way. So as these electrons go down these proteins, they pump H plus ions into the intermembrane space. And so what ends up happening is that these H plus ions will flow down in the one way they can because they don't want to be in high concentration relative to each other. They just want to be as spread out as they can, so they go the only place where they can go, which is through this protein called ATP synthase. And what this is going to do is H plus rushes down, ADP will combine with a phosphate group to form ATP. And this is where most of the ATP in cellular respiration is from. And this is kind of similar to a water wheel, as it's rotating as H plus ions are rushing through instead of a wheel rotating as water rushes through. Speaking of water, water is also formed in the electron transport chain along with the oxygen, the electrons from the FADH and NADH, and along with some hydrogen ions. They form this water here. So here's a net reaction. As you can see, we have 10 NADH and 2 FADH2 per glucose molecule. That's approximate, but you can pretty much assume this. Plus the 6O2 is going to lead to these guys losing their electrons and some of their hydrogen, and we're going to form 12 H2O. Once again, this is the H2O that we breathe out. So we've created. Let's see here. We've created 2 ATP in glycolysis, we've created 2 ATP in the citric acid cycle, and we've created about 32 ATP in oxidative phosphorylation. So that's a total of about 36 ATP per glucose molecule, and that's an approximation. Alright, let's go ahead and wrap this up by talking about where all of these molecules come into play. So first of all, we start with glucose. That's going to enter into glycolysis and form pyruvate, and then through a series of reactions, it's going to release CO2 and H2O. The H2O being released from the electron transport chain, and the CO2 being released from the citric acid cycle. This oxygen is necessary to pull the electrons down the electron transport chain. This creates a H plus gradient, and this in turn will drive ATP synthase and then make ATP, that 36 ATP. But what do organisms do if they don't use oxygen? Well, what they can do is they can loop through glycolysis. So one example of this is lactic acid fermentation. That's when glucose goes through glycolysis and forms pyruvate like any other form of glycolysis, and once again this forms 2 ATP, and this adds electrons to NAD+, forming NADH. But the problem is that without oxygen, this NADH will never turn into NAD plus without the electron transport chain, because oxygen's not pulling the electrons down. But what can end up happening is pyruvate can combine with NADH and then form lactate, and then this forms NAD plus, and this whole entire thing can loop over and over and over again, generating 2 ATP. This actually occurs when we are working out and when we're sprinting, because not only will we go through aerobic respiration or cellular respiration with oxygen, but we'll also be doing this as well, and this will be producing that extra 2 ATP. And that's like this lactic acid right here. This buildup actually causes us pain, so that's why it hurts when you work out. Okay. Alcoholic fermentation. Once again, same concept where we need to replenish this NADH from the or NAD plus from the NADH. But instead of forming lactic acid, we're going to form ethyl alcohol, which is a 2-carbon molecule, and so we'll also need to release a carbon dioxide here. Yeast go through this, and this carbon dioxide that gets released is actually what makes bread rise. And then once again, here's glycolysis. You have using 2 ATP to make 4. That's a net of 2 ATP for every single loop. and the formation of ethanol and lactate allow this NAD plus to be replenished. All right, and then finally we have anaerobic respiration, which is pretty much the same as aerobic respiration, except for the fact that oxygen is not the final electron acceptor in the electron transport chain. The electron transport chain still occurs, but a different molecule that's very attracted to electrons will be the final electron acceptor. So, example, we have SO4 2 minus, and then sulfur, and both of these will, instead of forming water, will form H2S. And I guess that's it.